Very own Associate Professor Mark Green, very own, I mean, the University of Melbourne. He's a reproductive biologist at the University of Melbourne and the Deputy Scientific Director at Monash IVF. His lab uses assisted reproductive technologies to determine how environmental factors such as emerging contaminants, heat stress, and light at night affect fertilities in humans and a broad range of species. In particular, whether chronic exposure to endocrine disrupting chemicals is responsible for an increasing rate of unexplained infertility and reproductive disorders in humans. So Mark, if you're there, if you could turn your camera on and take over the screen, please.
Hey Brad, I'm there. Can you, there we go. Can I share the screen? Is that all right?
Should be able to.
All right.
You'll be invited to the Alec Cafe post-talk and you'll need to tell me your favourite coffee, okay?
Pressure. Okay, yep, I like the pressure. All right, I'll keep that in mind as I'm presenting.
Good.
All right, so thanks, Brad, for the opportunity to talk today. And I'm, I guess, taking on the baton from where Gabby's left off in terms of looking at fertility, or at least some of those estrogenic and androgenic compounds. And really, I'm there as a biologist to maybe persuade you that also one of the other ways that we could look at this is to look at fertility itself in the animals, or in fact in the dish when we're thinking about eggs and sperm, and looking at how they can inform us on long-term health implications. So although we're talking really about emerging contaminants, I'm going to be focused specifically on endocrine disrupting chemicals. Firstly, because I think there are a number of compounds and families, of course, that we're well aware of. They're not new, but there are definitely new compounds within each class that are starting to appear on the scene. But also, don't forget, we don't know many things about some of their effects. So we might be able to measure them and quantify them, but really it's a bit of a black box in terms of where we're at. So we know that there's about 1,500 at the moment chemicals in the EDCs that have been defined to have some unwanted side effects from that side of things. And this, of course, is an ever-growing list. And on the screen are some of the key players that I guess you can understand and have been well aware of in terms of the bisphenols and the parabens and the phthalates, in terms of some of the short-term exposures that we can expect and their devastating effects on fertility. But following on from Chelsea's definition, I guess in terms of the POPs, they're probably the ones that we're more focused on in terms of their long-term effects on fertility. And some of those legacy ones, such as the PCBs, but also the big classic superfamilies such as the pesticides, herbicides, insecticides. And of course, more recently, and something that Brad and I have been working on, is looking at PFAS and their effects on fertility. And I guess from Chelsea's talk, we'd also put microplastics potentially in there. But then, of course, we shouldn't forget some other ones there in terms of air pollutants, and of course, whether it's in the developed or developing worlds, in terms of some of the pollutants that we know can affect fertility and through actually EDC effects. And of course, fracking itself in the last few decades has come online, and some of the compounds that have been released through that. And of course, historically from heavy metals, where we've seen that utilized in human industries and in terms of how they got into our environment. So really, it's just showing that although these are probably some of the main big players and their families, and we should consider them as emerging and emerged contaminants in terms of what we don't know and what we could know about their effects, and using fertility to understand these. So what do we know in terms of some of these EDCs? Of course, classically, we know in terms of specifically those animals and species that are living in the watercourse. So obviously the fish and amphibians there. We know, of course, some of the deformities that were anecdotally reported and then, of course, experimentally reported many decades ago. Leading on to, of course, some of the fertility issues we know in terms of alligators in the 1980s and 1990s. And then, of course, looking not just those that are in the water system, but those kind of apex predators at the top of the food chain. So whether they're aquatic in terms of those orcas, or in the air on the land in terms of bald eagles, for example, all of these that we know were through the bioaccumulation pathway of POPs and many other compounds, of course, have affected fertility in these species. So I guess we shouldn't see that we're better than these species, and we should think about humans as one of these species. And when we're considering EDCs, of course, we've heard, and we will hear, of course, today about how they can affect many different organ systems, everything from thyroid, metabolism, brain, all the way through to our bones. But of course, because a lot of these EDCs have unknown or multifactorial, multi-organ effects, then it's how do we actually study them and what is the best marker for that? And I'm going to suggest that one of the best things that we can look at is fertility. So why fertility? I guess it's really the fact that all of the other ones above the top there, of course, are ones that we absolutely need to survive, or any species needs to survive. But of course, fertility is something that is a luxury that we don't need to do. Of course, we need to, of course, reproduce. But in terms of me as an individual, I don't need to survive to have kids. So really what we're looking at then is when we're stressed or when things go wrong, fertility is obviously the first thing the body will usually shut, or at least stop, or at least decrease, or in some cases increase. So I guess it's a really good marker because it is the most sensitive probably of some of these systems, an early warning call, I guess. And so really what we're looking at is, can we use fertility as a proxy of health in all species, especially humans? So when it comes to humans, you probably think we're a little bit immune to this sort of thing, but we're absolutely not. So now there have been many studies looking at huge studies in terms of with 50,000 people, in this case men. So we're looking data here from a couple of studies I've picked out, and really looking at that association between semen characteristics, infertility and long life. And so from these studies now it's become quite clear that when we look en masse, we can see that sperm parameters are positively correlated with life expectancy. Looking at the data, of course, one of those confounders, of course, is whether these people, or these men in this case, have children. And of course, they found there was no association with the presence or absence of kids, or that anyone that's been homeschooling for the last few months, I dare to find that I've shortened my lifespan. But that said, they did find that basically that when you look at semen characteristics and fertility, it has a very, very high predictive ability in terms of the longevity of that individual. And it's so strong that actually it's equivalent to the risks for that of smoking and diabetes. And I don't think many people realize that fertility can be that useful in terms of a marker. So if we're not dying from homeschooling, then what are we dying from? And in our cases, when we're looking in terms of humans, then really we can see that what's underpinning the good semen quality, or in case of what would be expected in females in terms of egg quality, then it's really about the steroid hormones. And of course, we know that they're fundamental for many of our systems. And so actually we know that when we perturb these steroid hormones, for example, we get increases in the cancer incidence, specifically in some of those reproductive organs, such as the testes. We know that, of course, androgens are important for cardiovascular health and metabolic health. And of course, men are then leading on to dying from these. And then, of course, they're fundamental in our inflammation and inflammatory diseases. And we'll hear a little bit later, I think, in the workshop about those. So all up, what we're showing is that there is quite a strong relationship between fertility as that canary in the coal mine for health. And so using that, if you like, as a bioassay, once we've identified compounds, or if we're unsure about whether compounds have an effect, can be quite useful. So what are the effects? And if we think about humans for a minute here, you can see in the diagram, then really what I've got here are some of the major organ systems that we know are affected from a fertility point of view. Obviously the brain is probably a major organ in terms of sexual behavior, but all of these other organs down here in their own right, of course, are important in terms of effects of EDCs, but actually indirectly can affect fertility through immune responses, sensitive tissues for cancer, and of course, obesity, well known to decrease fertility. When we look at the actual reproductive tract, it doesn't matter whether it's male or female. So from the male side, we know that there's issues in terms of testis morphology and function, penis characteristics, and some of the diseases and disorders associated with them. So hyperspadias is one of the most common birth defects here in Victoria, and in fact across Australia, with 1 in 120 male babies being born with hyperspadias. And that is basically where the urethra doesn't open, doesn't end at the end of the penis, and it actually can open anywhere down the shaft of the penis. So that is very, very prevalent and increasing in incidence, and something I'll touch on in the last few slides. In terms of females, then we know equivalents in terms of the ovarian function. So polycystic ovarian syndrome, or PCOS, endometriosis and cancer, all very, very highly prevalent, and with PCOS actually being up to about 14% in the Australian population of fertile women. Of course, when we do manage to conceive, we shouldn't forget that EDCs are going to affect our fertility in terms of pregnancy. So when you know that we're going to see characteristic changes in the fetal growth, in preterm incidence, and sadly with stillbirth. So we can see across the board in humans, of course, that we can see fertility being affected by exposure to emerging contaminants such as EDCs. So those were in the sort of association to diseases and very severe disorders. But what wasn't really covered well was really if around those I guess, people that are trying to conceive and around the time of conception, what were the direct effects at that time in the life stages? And so our recent review, we looked at a number of studies, in fact 350 studies, to see what kind of strong evidence there was out there in terms of EDCs looking for fertility and fecundity. And when we look for the male, then we can see that actually there is strong evidence for decreased sperm motility, increased DNA damage through exposure to PCBs and pesticides. Equally for the female, we see changes in the menstrual cycle length, decrease in estrogen, and of course egg number and quality is affected as well, again by things like BPA and PCBs. For those that do try and conceive, then the time to conceive is increased through things, again, through PCBs and BPA and phthalates, and increasing miscarriage rate. So that's some of the very strong evidence that's currently out there. On top of that, there's lots and lots of evidence for other compounds and emerging contaminants in terms of some of the characteristics. So what was obviously apparent was actually when we do review the literature in terms of what we know, even as humans specifically, there are actually very few studies, especially in females. And that's, of course, because access to eggs, of course, is not as easy as it would be to sperm samples. And so actually there is a hole in the literature, the gap in that knowledge in terms of what's happening with emerging contaminants in female reproduction. Also, it was apparent that really when there was no statement, people often assume that obviously it's safe, but that's not actually the case. More often than not, it's because it's not been studied. And so we need to be careful to remove that bias when we're thinking about possible effects. So what are we doing in terms of this area, in terms of using fertility and research studies on EDCs and other emerging contaminants? So in my lab and others, we use a number of different species in terms of rodents, classic, of course, workhorse of the biomedical industry. Domestic species, due to their size, such as cattle, and similarities to some of the reproductive characteristics, wildlife, and of course humans. And I'm currently conducting many studies in Monash IVF of patients coming through and looking at associations to their reproductive outcomes. So really the point is that we want to delineate the direct effects on the eggs and the sperm that then of course make the embryo and then the embryo itself. And so we can understand how some of these compounds, emerging contaminants, can actually affect them using biological endpoints. We also want to have a look at the whole animal implications. So when we actually are looking at the animal itself, how the maternal tract, for example, is engaging with the embryo and the pregnancy and the fetal growth, and also the incidence of reproductive disorders. Within that, of course, we know that through many years, in fact, the last 30 years, we know the developmental origins of adult disease. We know that actually things and exposures or insults around the time of conception, both on the egg or the embryo or the sperm, can actually have lifelong consequences in terms of disease. And so we see that this programming of disease occurs right around conception. So it's very important that we understand what's happening specifically around that time point. And as shown in this diagram, we know that, of course, if you are a pregnant female, for example, of any species, then what you're actually getting is the emergent contaminant, of course, is being exposure to mum, but also to baby. And of course, within baby are the gonads or the ovaries or the testes that will become the next generation. So we do get multiple generations being exposed at once when we're considering that. And so we need to think about the long-term consequences of that exposure. And that, of course, comes in the fact that we know that these EDCs, or in fact any contaminant, can affect and set thresholds, for example, for that embryo and that developing fetus in terms of predicting the lifestyle and the outcome once they're born. And it does really set them, unfortunately, onto a course to mean that they are actually more disposed or predisposed to risk for things like obesity, cancer, for example, which themselves, of course, can negatively affect fertility. And of course, this manifests all the way through to the next generation, affecting reproduction. And unfortunately, we have this perpetual cycle in which we're ever living in this soup of emerging contaminants. So it's really important to understand how this is really impacting and confounding over generations. So my lab has really focused on a number of these EDCs, for example, first of all, starting with bisphenols and showing that actually even concentrations that are environmentally relevant can affect the embryo development and metabolism, even from a very short duration in exposure. And now more recently been focusing heavily on atrazine, which of course is the second most prevalent herbicide used in Australia and in fact many countries, and looking at its effects directly on the early embryo, but more specifically on the male side of things and that incidence of hyperspadias, which is that penis deformity. We have also gone on and looked and currently looking at the effects of parabens, for example, in multi and transgenerational models in the female side. And in both animals and humans, we're currently looking at the effects of PFAS and how these in populations of males who are living around departmental defence bases around Australia are affecting potentially using fertility as effects on multi-organ systems. More recently, and highlighted, I guess, nicely by the last talk from Gabby, we need to understand how mixtures are working. And so whilst we often think that having agonist effects, they can have interactions and antagonists with the mixtures. And so it's important to really have a look at how these mixtures work when they're put in an in vivo and in vitro setting. And that's something that we're starting to do in multi and transgenerational studies. So I'm just going to give you some example of how we can use fertility, and specifically I'm going to use atrazine and look at the effects on male fertility. So of course atrazine is well known. In fact, it's one of the few EDCs, emerging contaminants, that does actually have a so-called safe level in Australian water system, for example. And we know from Tyrone Hayes and many other people's works that it then of course causes feminization in amphibians and fish, and then subsequently from animal and in vitro studies, the effects on endocrine and metabolism. So historically, of course, a lot of these studies have been very high dose, but we wanted to look at what happens with the environmental so-called safe levels. And we used the mouse model to look at this. And we found when you're looking at these very, very low concentrations in the environment, then of course, you're not going to find things such as testis morphology changes. But when you look at more subtle parameters, such as sperm characters, we see these decrease, and we see altered hormones and testis concentrations. More importantly, we did see differences in terms of the penis defects. We found that those males that were exposed to atrazine had smaller penises, as shown in the graph on the top hand side here. And when we did histology, you can see, or you may not be able to see, but this is actually a section through the penis itself, and this is the control on this side, the atrazine on that side, and that's right at the top of the penis. But as we go down the shaft, what should happen is that we should lose that opening, and of course it maintains in that atrazine, showing that hyperspadias syndrome that we see. So we're able to see by looking more closely at some of the subtle effects at environmental levels, that we can see that things like atrazine can cause penis defect and increase in hyperspadias, which of course is commonly reported in humans. So we then look at transgenerational, multi-generational effects in paternal and looking at breeding those animals, and we've done that. And when we've exposed them to unexposed females, that again, we don't see dramatic changes in the embryo development, but when we look more closely at whether these diseases are being programmed in there, we can look at measures such as the number of embryos, number of cells within the embryo, or its metabolism. And we can do that by counting the cells within the embryo. And so this is a mouse embryo, and the blue represents the inner cell mass that becomes the fetus. And the pinky purple is the outer cells, the trophectoderm, that become the placenta. So we know that changes in these ratios lead to programming through many, many different diseases and disorders. And actually we found that when we did look at this, we can see that the inner cell mass, while the fetal numbers don't change, actually the cells that become the placenta, and we see reduction with atrazine and in fact total cell number, and showing that we can have this transgenerational effect. So in summary, then what are we doing in terms of fertility and long-term health, and what can we do in this space? It's important to think about multi versus transgenerational models that we can use, and even in wildlife. And of course, classically, we've really focused on multi-generational with single exposure, and then following them through to the F2. Whereas transgenerational, of course, it's been a single exposure and then watching them pass the F3 once they've been passed on. Really, what we need to know now is really what multi-generational effects are happening when we're having continual chronic exposure over multiple generations, because that's exactly what we're living in and observing that for generations, and that's the work that we're currently undertaking. Of course, in humans and wildlife, then we're looking that it's a good proxy for health because of the association with decreased fertility, increased diseases, with now 1 in 6 humans needing help in order to reproduce in most Western countries. And for future research, then really it's important that we agree on how to look at these subtle effects and methodologies and endpoints for both those that we consider emerged contaminants, as well as emerging, and more importantly, the mixtures that we're seeing and being identified. And really how we use this in terms of long-term multi-generational and transgenerational effects to look at numerous diseases. So hopefully I've just shown you or given you a hint that we can actually use fertility to gauge the multi and transgenerational effects of emerging contaminants. Thank you.